Yes, it is enabled. Okay. Molecular docking, as I said, is an example for structure-based drug design, and it is very commonly used protocol. Even if you take the top pharma companies, even international pharma companies, the computational chemistry group of those who are involved in the drug discovery process, they used to do molecular docking. So every month you can see, several hundreds of papers in which they have used molecular docking as one of the protocol for structure-based drug designing, right? So why it is so popular? Because this method is very promising, right? So there are many molecules which are discovered with the help of molecular docking, right? Uh, so let me start with a very fundamental thing. I just wanted to tell what is meant by molecular docking. So this is something like making a complex between two molecules. A simple way to define what is meant by molecular docking. Dock. The term literal meaning of dock is connecting two components, right? In drug discovery, the molecular docking is say A plus B, which gives a complex A B. The formation of this A B complex with the help of computer algorithm. So let me say this is complex. The formation of this complex with the help of computer algorithm by using the molecule A and B. That procedure or protocol is what is called as molecular docking or molecular docking studies, right? So why this protocol is important? How this is related to drug design or drug discovery? Because we in detail discussed about what is meant by drug and drug target. We also discussed about the drug action, the relationship between the drug molecule and the drug target. So the drug molecule should go and bind to receptor, corresponding receptor, where the drug molecule and the receptor should interact with each other to form a drug receptor complex, and the formation of that drug receptor complex may lead for the biological response. So this is what we call it as the drug action. Okay, so the formation of this complex is very important. Okay, that lead for the biological response, and there are different ways by which we can make this complex, right? So we can do some wet lab analysis, but this is simplest thing, right? So using the computer we can form the complex. Okay, this procedure or the protocol is what is called as molecular docking, right? So what for this is used? Say for example, this will the if you go for the molecular docking study, to some extent we can accelerate the drug discovery process. So let me give an example for why normally we go for molecular docking. Why it is very famous, right? Say for example, I have set of ligand molecules. I have set of ligand molecules with different shape, right? So I have 4 ligand molecules, and this is my target receptor. Okay, so say for example, if I go with the wet lab technique, what I have to do is I have to take all the 4 molecules, and one by one I have to treat to the receptor, and I have to check the affinity of the each ligand molecule or the inhibitory or the activation of the ligand molecules for the corresponding receptor. Okay, so if I go for the wet lab technique, what I literally should do is I have to take the first one and check whether the molecule is working. Take the second molecule and check. Take the third molecule and check, and take the fourth molecule and check. Right. So if I do this experiment in wet lab, okay. So for which molecule I'll get the result? Say you are working for finding the inhibitor for this receptor. Which of the 4 molecule do you think can inhibit this receptor? Sorry.
First one.
Okay, if I number this 1, 2, 3, 4, I don't know who said, but the answer is right. The first one, we can expect that the first molecule will show some activity. Definitely not the second or third or fourth, right? So can you tell how do you say this?
Shape
How?
complement.
Okay, so what we need is basically we need a kind of shape complement, right? So that we cannot even imagine if we go for wet lab analysis, right? So the amount spent, right? So we have to buy the first compound, second compound, third compound, fourth compound. Finally, only one compound is showing activity. If I follow the wet lab procedure, right? But instead of that, if I take all the four molecules and if I try to make complex between all the four molecules one by one with the given receptor using the molecular docking algorithm. Okay, so the drug molecule plus the receptor. So each molecule here is a drug-like molecule. So one by one we have to dock. So which molecules do you think can be docked in this receptor? Can I dock the second molecule? Can I dock the second molecule? Or can I
No,
dock the
sir.
third molecule?
No, sir.
Can I dock the fourth molecule? But I can dock the first molecule, right?
Yes, sir.
So the output is that the computer algorithm will tell, okay, the first molecule, there is a shape complementary, the molecule is able to bind. But the second molecule or the third molecule or the fourth molecule, the computer algorithm itself will tell there is no shape complementary. So there is no affinity between the second molecule, third molecule, fourth molecule to the receptor of our interest. So the user can conclude that, okay, I want to purchase only the first molecule. I should not purchase the second, third, or fourth. Okay, so two ways this makes sense. The first is that it's going to reduce the money. Definitely, I'm not going to purchase all the four compound. Secondly, it will definitely reduce the time for the wet lab techniques because they are time consuming experiments. That's the way how we can make use of the molecular docking. So any doubt up to this what I have discussed, I can address. If not, I just want to proceed to the next slide. Any doubt up to this? Just I introduced what is meant by molecular docking. That's all. If you have any doubt, I can answer at this point. You people can respond.
No, sir.
No doubt. Okay, fine. So, okay. So what we can infer out of any molecular docking studies is that the molecular docking study will suggest the binding mode of the drug molecule or the ligand molecule of your interest. So each ligand molecule can be positioned or we call it as pose. There are possibility for the same ligand molecule to be fitted in different pose, right? So I just want to give another example for this pose. Say again, I am taking the same architecture, binding pocket architecture. So this time I just wanted to use three different colors. So say uh, yeah. So this is my first molecule. Okay, this is my ligand molecule, and yes. Okay, this is my ligand molecule. Can I fit this ligand inside the binding site now? Can I fit this ligand molecule inside the binding site?
Yes, sir.
Yes, right. So even if so what will happen? The green color will come to this position. Am I right? The green color will come to this position. The red color will go to this position, and the violet color will present in the top. Am I right? You will get something like this, right? Am I right?
Yes, sir.
Okay, so now can I rotate this molecule and fit the same molecule inside the binding site? So can I rotate this molecule like that? The violet color come to different position. Okay, say the violet color is now present here. the red color is present here, and the green color is present here. Can I fit this molecule now inside the binding site or not?
Yes, sir.
Yes, right. Both the molecules are same or different?
Same in shape.
Same in shape or same? Both the molecules are same or not?
Yes, sir.
So both the molecules are shape. Everyone agree with that? Please respond. Right? Okay.
Yes, sir.
Yes, right. So both the molecules here, what I have drawn to illustrate is same. But can I say different orientation?
Yes, sir.
Yes,
Yes,
right.
sir.
So the same molecule can be fitted inside the binding site in many number of orientations. So each orientation is called as a pose. So what I mean here is each ligand molecule can be docked inside the binding site of the protein molecule in many number of poses. Okay, but not all the poses will have the same affinity, or all the poses will result in the same energy. Maybe a bit confusing for you. So I just wanted to give some more clarity in what I mean here. So I'm taking the same example here. So now I am going to make a small change at the binding site level. Say for example, here you have a positive charge. Here you have some amino acid with negative charge. Here you have some amino acid with negative charge. Okay, so so here you have a negative charge. Here you have a positive charge. Here you have a positive charge. Okay, in the second case, say for example, just for example, right? So here you have a negative charge. Here you have a positive charge. Here you have a negative charge. Okay. So now I hope the visual is clear here. We have two molecules. Okay. Now we have some charge on both the protein as well as in the ligand molecule. So which of the two orientation is having the exact stereoelectronic complementary? So the meaning of stereoelectronic complementary is the positive charge should interact with negative charge, and the negative charge should interact with positive charge. Can I say the first conformation is having very good stereoelectronic complementary?
Yes, sir.
So I can say
Yes, sir.
that the first pose is having both shape complementary and perfect stereoelectronic complementary. So coming to the second conformation, both the molecules are same. I can say that the second molecule is having good shape complementary, but the stereoelectronic complementary is not good. Do you agree? Am I right or wrong? So here there is a negative charge. Here there is a positive charge. There is a negative charge. So these two sides will attract, but this side will repel at this position. Am I right? Negative, negative.
Yes, sir.
So which one will be having higher affinity now? Molecule 1 or molecule 2?
Molecule 1.
Molecule 1, sir.
Molecule 1 will have a very high affinity comparing to molecule 2. The reason is it's having both shape complementary and stereoelectronic complementary. But molecule 1 and 2 are same. Only the orientation or the conformation is different. So just like that, if you dock any small molecule in the binding site of the protein molecule, the small molecule can be kept inside the binding site in different orientation. But not all the orientations will have the same affinity. The affinity of each orientation will differ. It depends on both the shape complementary and the stereoelectronic complementary. Okay, so if I say that we can position the ligand molecule in different ways inside the binding site, there should be some way to tell that which conformation or which pose is the best pose. So in our example, the first pose is the best pose. So if I rank 1 and 2, the 1 will get the first rank, and the second conformation or the pose or the orientation will go to the second position. So if you have any molecular docking algorithm, okay, the algorithm could able to score each structure to identify the most interesting pose or the conformation. Okay, these are very basic fundamental things in molecular docking. So this is the first definition given for molecular docking in 1993. Okay, this is not new story, or it's not new technology, right? So molecular docking is, it's having its own, history. Sorry. So the docking problem is thus concerned with the generation of evaluation of plausible structures of intermolecular complexes. So we have to form the complex, and we have to generate different number of conformations within the binding site for the given drug molecule or drug-like molecule. And the algorithm could also able to find out the intermolecular interactions which lead for formation of the complex, right? So I'll explain this in detail, no need to worry about this. So this slide is the most interesting slide if you're interested in designing some program for molecular docking. It's not so difficult, also it is not simple, but you can also develop a software that can perform molecular docking. Why I say this? Because the biology. Bioinformatician, they know the biology thoroughly. Also, they know how to write program. So if you have the knowledge of biology and knowledge, very very strong knowledge in writing program, anybody can write the docking program, right? So what I mean is that we say autodock, we say glide docking, we say flexex, right? Even you can write some program like that if you know the basics of molecular docking and the complete thorough knowledge about biology and writing program, right? So whenever we develop any algorithm, or we learn any algorithm, or we learn any software, so in bioinformatics, first thing is we have to create the hypothesis, and we have to define what is the problem which is associated with that what we are working on, right? So here when we talk about docking problem, so the first thing what we have to think, right? So before writing a program or before understanding the functioning of already existing algorithm, so any docking program which involves many degrees of freedom, maybe almost not all, a few terms may be very new for you. So do you know what is meant by degrees of freedom? Any paper, any coursework have you studied about degrees of freedom? If I say DOF or degrees of freedom, if you respond quickly, I can I can move a little fast. If you,
How many external factors are affecting its change?
how many
External forces or external agents that are affecting it to change.
okay, so okay, any other answer? Any other way to tell what is meant by degrees of freedom?
In how many possible ways a molecule can take the conformation?
Okay, so both are somewhere right. So that is what is meant by degrees of freedom. So when we think about molecular docking, so there are mainly 6 degrees of freedom. Okay, so among the 6 degrees of freedom, 6 DOF, 3 degrees of freedom corresponding for translation, and 3 degrees of freedom corresponding for rotational freedom. So what is meant by translational and rotational freedom of a molecule? Say for example, if you have some molecule like this is your drug molecule or ligand molecule. So what I can do is I can move this molecule in X direction, I can move this molecule in Y direction, or I can move this molecule in z direction. There are three possible. In the three-dimensional space, I can move this molecule in x direction or y direction or z direction. That need for three of the translational degrees of freedom. That's called as translation. Moving the molecule in x direction, y direction, and z direction. So one more action what I can perform on this molecule is I can rotate the molecule. Physically, I can rotate the molecule. So I can rotate again the molecule in the x-axis. Is my screen visible now?
Yes, sir.
Am I audible? Yeah. I don't know when I disconnected. Can you tell what exactly I discussed before disconnection?
Rotational translation.
I don't know when I, okay, so 3 degrees of rotational and translational degrees of freedom. Okay, so the simplest algorithm will handle this 3 rotational and translational degrees of freedom. As I said, that anybody, even you can write a program that can handle that. So in this, what we assume is both the protein molecule and the ligand molecule, they are highly rigid. There is no flexibility for protein and there is no flexibility for the ligand molecule. So the meaning of flexibility here is there is no conformational change that is possible for the ligand molecule either or the protein molecule. But naturally, both the protein molecule and the ligand molecule, they are highly flexible. So can you tell which molecular recognition theory that assume both the ligand molecule and protein molecule as a rigid body? Because this we discussed in while talking about stereochemistry. If I remember, I have shown 3 theories, and one of the theory which explains the nature of flexibility and rigidity. Do you remember? Am I audible?
Yes, sir.
Yeah, you can answer. I am not expecting yes, sir. Yes, sir is no answer. So we discussed about lock and key theory. We discussed about fit theory. We discussed about 3-point theory, if you remember, right? So which of this theory assume that both the protein and ligand molecules are highly rigid? I mean, completely rigid. There is no which of this theory assume that both the protein and ligand molecules are rigid body? There is no flexibility. I'm expecting some answer. Is my question understandable?
Sir, can I
Sir, lock and
question?
Am I audible?
yes, sir.
Sorry? Lock and key, right? Lock and key model, right? So any basic docking algorithm which takes the concept of lock and key model, molecular recognition, because that is a theory that state that both the both protein molecule and the ligand molecules are highly rigid. Okay, rigid body. But we have some docking algorithms which are very complex in nature. They can handle the conformational degrees of freedom. Okay, so the meaning of conformational degrees of freedom is if you have a carbon-carbon single bond, and wherever you have the carbon-carbon single bond, either in the protein molecule or in the ligand molecule, they can undergo rotation, which lead for many number of conformations of both the ligand molecule and the protein molecule. So that is what is called as conformational degrees of freedom. So once if you incorporate the conformational degrees of freedom, which means that you are allowing the molecule to undergo conformational change during the docking simulation. Okay. So as we know that always the structure of uh, drug targets are very large comparing to the structure of drug molecules. So we can to some extent manage the conformational flexibility of the ligand molecule, but absolutely that is not possible for we cannot give the conformational flexibility completely for the protein molecule, because in average the drug target may be made up of 200 to 250 amino acid average, right? So there's going to be many number of carbon single bond, because each amino acid it has at least a few numbers of carbon single bond. Eventually that lead for, if you add all the bonds, definitely there is going to be large number of carbon single bond, and giving flexibility for the drug molecule the giving the flexibility for the macromolecule or the drug target, it's going to be very challenging. But the point to remember is, whenever you write any docking algorithm, so you have to think of conformational flexibility, because naturally both the protein molecule and the ligand molecules are highly flexible in nature. Okay. So basically we have two types of docking programs. So the first class of docking program is called as automatic docking program. Second class is called as manual. As the name indicates that the first one is very user friendly. So all the parameters are almost controlled by the algorithm itself, the software itself. The only thing is the end user has to input the protein and the ligand molecule, that's all. So all other things will be handled by the software itself. Those type of algorithms are called as automatic algorithms. And there are few manual algorithms in which the user take the complete control. The user has to define whether the simulation is going to be a flexible or it's going to be a rigid body kind of simulation. So if it is going to be flexible, so which amino acids are going to be flexible, and which atoms in which bond in the drug-like molecule is going to undergo conformational change during the simulation. So what I mean here is, everything the user is going to control in case of manual, but that will be taken care by automatic algorithm. So if we which one do you think is good, automatic or manual? Which algorithm do you think is good? A kind of automatic algorithms or manual algorithm?
Automatic, sir.
Okay, automatic, do you think is good, right? So, but not right. Okay, always it is better to go for the manual, because the user has the complete control. Any computer algorithm is just mathematical, flow, right? So, mathematical formula and kind of flow, right? So, if you are really experienced, then obviously you should go for only manual, not for automatic. Automatic is just for the beginners, not for expert or advanced. They will look for controlling all the parameters. They won't allow the algorithm to take care of everything. They will try to control everything. Okay, then only you can understand what exactly is going on. Okay, so that's the difference between automatic and manual. So coming to the examples for docking algorithm, so we have a program called as UCSF dock. Okay, it's a very famous docking algorithm program. So we have autodock. Almost everyone you may already used, or you may heard about this. A very commonly used docking algorithm. So we also have FlexX. FlexX. We have Glide. We have a license for this. We have license for this. And gold is another famous docking algorithm. We can give many name, right? So nowadays, there are a lot of docking algorithms. But anyway, these are very we can say that very commonly used, still successful molecular docking algorithms. And some of these docking algorithms are simple algorithm, and some of these docking algorithms are a very complex, advanced docking algorithm. Say for example, if we take a dock, UCS of dock, we can say to some extent it is a simple docking algorithm, because it assumes both the protein and ligand molecule as either the protein molecule or the rigid molecule, right? So we can say dock, UCS of dock is a kind of completely rigid docking algorithm, a kind of basic algorithm. But on the another hand, if you go for FlexX, we can say it's a kind of advanced docking algorithm. Same thing, Glide also kind of advanced docking algorithm. So in the sense, the user can give flexibility for the entire ligand molecule, and to some extent we can give flexibility for the binding site amino acid. There are cases in which even we can give maximum number of amino acids as a flexible residue. So this autodock is kind of semi, which lies in between the rigid docking and the advanced flexible docking algorithm. So using this autodock, we can give flexibility for a few number of amino acids within the binding site, and we can make the entire ligand molecule as completely flexible. Okay, so this is how we can classify the docking algorithms into simple, complex, or way, right? So these are very successful docking algorithms. Any question up to this I can answer. If not, I'd like to go to the next slide.
No, sir.
Yeah, I know that the answer is going to be no. Okay. Okay. So the first the software what I have mentioned. We'll look into the actual algorithm part, or the working principle of doc, UCS of doc. Why we choose this? Because as a beginner, it will be easy for us to understand how this docking algorithm is working, because this consider both the protein molecule and the ligand molecule as a rigid body, right? So this will give some idea or picture about how they have developed the software, and if you want to develop any software, what methodology you have to follow, right? So so for giving some idea about that, so we have chosen this UCS of doc algorithm. And almost all the algorithms will have a few steps which are very common, and we can say that irrespective of the software, the first step of docking any algorithm, right, so is going to be the same. So the very first step is we should look for structure solved by X-ray crystallography. So this is what we have discussed in the previous lecture, the role of X-ray crystallography in structure-based drug design, right? So as molecular docking is an example for structure-based drug design, the very first step is we should know the 3D structure of the drug target. And the best way to solve the 3D structure of the drug target is going for X-ray crystallography. We can also go for NMR and electron microscopy, but this is the very preferred method for drug design. Already I told the reasons, right? First step is, once if you have the 3D structure of the drug target, so very next step is we had to identify the amino acid lining the binding site. Okay, so if we can simplify this, the very next step is we have to identify the binding site. Okay, say for example, if this is your protein molecule, and if I ask you a question, where is the binding site? What will be your answer? Where is the binding site in this protein? Just a graphical illustration. So how will you identify where the binding site is present? Which methods you will follow? I asked this question assuming that you completed your structural bioinformatics. Again, I repeat the question. I have drawn a kind of illustration of a protein molecule. You have to assume that it is a protein molecule. The question of interest is where the binding site is present in that protein.
Hydrophobic pocket.
Yeah, everyone know hydrophobic pocket. Where that hydrophobic pocket is present? How can I identify where the hydrophobic pocket is present? Is it going to be only one pocket, or the protein is going to have many numbers of such pockets?
Solvent accessible surface areas.
Who's that?
Rachana, sir.
Very good answer. Okay, so we have to calculate solvent accessible surface area. So the question is, how will you calculate the solvent accessible surface area? If I give any protein molecule, if I give the 3D structure of the protein molecule, how will you identify the solvent accessible surface area?
A water probe is allowed to run along the surface of the proteins.
Who is answering?
Rachana, sir.
Okay, absolutely right. Okay, I had to appreciate her answer. So we had to run a probe, right? So that's what she is saying, like water molecule. Okay, so we generally call this as a probe. Okay, not necessarily it should be water molecule, but it should have some characteristic, that's all, right? So we can generalize this as a probe. So we have to run the probe molecule. So first thing is we have to define the molecular surface of the binding site. Okay, whenever you have the three-dimensional structure of the protein molecule, first step is we have to define the molecular surface of the binding site. Hope you know how to visualize the molecular surface and what's the basic principle of viewing the molecular surface of the protein molecule. I hope you learned all those in at least a few softwares like PyMol or Chimera, right? So what we have to do is we have to identify each atom in the binding site. So for example, if this is the binding site of the protein molecule, so we have to identify each atom which are present in the binding site, and we have to represent that those atoms as its van der Waals radius. Okay, so here I have given one example in which each of this gray color sphere is van der Waals radius of that atom. That is how to understand this. Okay, so this is the way how we can represent the molecule surface of the binding site. So each of this, again I repeat, each of this atom represented as its van der Waals radius. Okay, so once if you have this once if you have this molecular surface, right? So this is what we do in the first step. We identify the molecular surface. I just highlight this. So this is what I mean here is the molecular surface of the binding site, not the entire protein molecule. This is a binding site. Okay, and this is a molecular surface of the binding site. And as she said that we have to calculate the solvent accessible area. Okay, so using the calculation of solvent accessible area, we can predict the binding site, exact binding site. So that is what somebody already told us, the hydrophobic pocket. So the hydrophobic pocket can be find with the help of this solvent accessible surface area calculation. Okay, so what we need is we need a probe molecule. So once if you know where the binding site is present, the next step we need a probe molecule, probe atom. So here the probe atom is a sphere of 1.4 to 1.5 angstrom. And if you look at this size, okay, 1.4 to 1.5 angstrom sphere, and that will correspond for water molecule. Okay, so that's why we call this as running the water molecule, or some probe molecule which is having a probe atom which is having 1.4 to 1.5 angstrom, right? So it should be rolled over the surface that we have identified in the previous step. And I just wanted to remove the annotation what I have made here. Yeah. So now this is very clear, and you can see how the water molecule is running. Okay, so we have to find out where the water molecule or the probe atom is able to make contact with the surface of the binding site. That is tracked here. Here you can see some dotted line. Okay, I don't know whether it is clearly visible or not. So these are the dotted lines. Okay, so which defines. Okay, so the places where the probe atom makes direct contact, or how it rolled from 1.2 to 1 can go. Beyond that, it cannot go. The meaning is you have a small cavity, but unfortunately, the probe atom, the water, cannot enter into this because the size of the probe is 1.4 to 1.5 angstrom. So this distance could be smaller than 1.4 to 1.5. That's why the water molecule cannot reach this, and the trace is moving like this. Is it understandable? Am I audible? Okay, so I just wanted to wind up, right? So I think time up. I'm not so happy with your class. There is no, answer from your end. I go very slowly because there is no interaction or response from the other end, right? So anyway, I just wanted to wind up the session. Thank you.